Hello, everyone. How are you? Let's take care of this first. I know it's a little bit early before it's 2 minutes before 3 o'clock, but let's take care of this business first. I thought about it and like to. If I only can decide on my own without getting any opinion from yours, I would like to change this whole exam date or push back several days to all Monday exams. There are two reasons why I want to do it. One is all Wednesday exams, but still that week we have to have the class. That means on Wednesday exam, that means on Monday lecture content will also be on the exam, which is like you have only 1 day in between, 1 day window to digest. So I don't think that's, I mean, if I were in your shoes, I would hate it. I mean, I just had a lecture the day before yesterday and it will be on the upcoming test. I would hate it. So that's one reason. So that if we just push this exam date until after the weekend of the lecture, then probably you would have several days more of the study for studying and digesting it until you come to the exam ready. So that's one reason. Another reason is by doing so, having the final exam only, having our final exam scheduled on 16 instead of the original, when was the 12th or 11th or whatever, then we could automatically take care of the one more, the Bogang left. I have uploaded this whole bunch of series of lectures, but that's only for those Chuseok holidays makeup exam. So what about another, the to be missed class, like on this coming Wednesday? There won't be any class either on Wednesday. So that one I have to also provide the makeup lecture. But the university rules are very stupid, but we are not allowed to more than 2 lectures of pre-recorded makeup lecture. So that means I have to offer the live lecture for that coming the makeup. No matter in somehow I have to improvise. So that is something that is not really convenient for me as well as for you too. But if I have this exam on December 16th, then automatically one more lecture. We are going to have one more lecture so that all this makeup lecture is taken care of. That's the two reasons I thought about how about moving this exam date onto these. Originally, it was like on Wednesdays or before, like on previous weeks. So what do you think? Would it be okay? Or are there anyone who, no, I can. There's a really, really very good reason that I cannot, for the life of me, I cannot take the exam for these particular days. And please raise your hand. Then we have to talk about to see if any better solution. No one? Are you sure? So then would it be okay for me to announce this changes the schedule of the midterm on snowboard? Would be fine? So the exam will be the upcoming midterm exam will be October 28th instead of 20 something, was it 23rd or I don't know, I don't remember. Okay, will it be? You're not. At least I can notice that someone is not really happy about this change. Are you? What's your problem? Oh, you have a plan to travel. You can talk to me. You can talk to me because you are, anyhow, you are not going to be graded on the regular the scale. So probably we can just talk about it and you can come up something about it. Okay? So see me after class. Okay, so anything else? All right. It was me. Okay, ScreenFlow is requesting to bypass the system private window. So what's your problem? Open system setting. What? I don't understand. I hope recording is doing okay. Let's check. Okay, so next thing is then let us quickly finish the chapter 2, and then we talk about briefly what to study on your makeup lecture, those chapter 3. As I promised, that I'm going to give you what specific topics, several topics that you need to study for your midterm exam on that chapter. And then we are going to move on to chapter 4 today. Okay. So this was the issue that one of the advantages of using mitochondria for comparing the evolutionary relationship among the different humans, as well as those humans, related humans that long extinct. So, for example, Neanderthals and between Neanderthals and other extinct the humans, ancient human, which one of the two was much closer to our direct ancestor, which was Homo sapiens type of question. To do so, I said on last lecture that DNA sequence comparison is probably the most accurate measure. Even we can have approximate estimate, pretty much accurate estimate of, the time scale from which these two different individual or two different species of humans got separated, or even we can accurately calculate. What is the so before we even go into why mitochondria and human Y chromosome in particular are the two of the best material for such task, we have to understand that why the DNA sequence comparison is why such comparison can provide such an accurate measure estimate of such evolutionary relationship among different organisms. It doesn't even only relate with these different humans, but it gets involved in all entire different organisms for such similar tasks. Like setting up correct evolutionary relationship by using DNA sequence differences is very valid, accurate technique and measure. Why? Anybody who can explain instead of me? Instead of my boring, awkward explanation, is there anybody else who can actually save you to explain this? Why this DNA sequence comparison can be such a good way of providing such information, evolutionary information. Okay, I give you, if someone who can provide this correct, very correct answer, then I will offer you 2 points. Not even 1 point, this time it's a 2 points. Midterm is coming soon, ladies and gentlemen. By the way, how was your bike trip? Was it good? Great. 2 points. Are you tempted? Okay. Why the DNA sequence comparison is such an accurate measure for comparing evolutionary relationship among different organisms. Like, for example, if you compare DNA sequence among human, chimpanzee, and dogs, the differences, like obviously we can anticipate that our DNA sequence is much closer to chimpanzee than to the dogs. Then by that, we can assume that our closer relatives is a chimpanzee rather than dog. Why? Why? Because of DNA sequences are so similar, then more distance is similar. Why does that mean that we are evolutionarily more closely related than the other? Is the question. Did everyone get that? You can even try in Korean if you are. Yeah. Yes. Oh, of course, sure. Everybody can understand Korean here. Uh-huh. Uh-huh. Hmm. 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 Okay, what she said, as everyone already understood, what she said was since the DNA sequence similarity means a similar protein as a gene product, so that means uh, just like the more you look alike, probably you can assume that you guys are more tightly related. So having just by virtue of having similar protein, that means you are more closely related evolutionarily. The answer is no. That is not necessarily true. Okay, you can assume, and a lot of times it may. Luckily, you may actually hit the jackpot by just simply following such logic. You may be able to get the correct answer, but that is not really actually the reason why we can trust this DNA sequence comparison as one of the golden rule of you can randomly. I mean, how do you know that we are totally unrelated, but just by chance event you could have a similar protein? What do you say then? So that is not actually reason. It's because the okay. So I think I gave you enough opportunity, so I will just close this. This is all about the common ancestor thing. So evolution is based upon this whole premise. We all derived from a single common ancestor, right? And what was the reason? If that was the case, and then what was the reason that we are all different by now? Exactly, mutation. Yeah, this mutation is the ultimate source of variation, right? But what's the characteristically, what's the nature of a mutation? Random. Mutation occur randomly. Anything that occur randomly is simply is the function of what? Time. In other words, the more you have, like the wider difference between individual A and B means more mutation, right, than the other case. That means these two individuals have been separated for such a longer time, since they are much in the longer past time-wise, is theoretically that's what indicates. Let me give you an example of identical twin. Let's say you're an identical twin. Identical twins are simply clone. Hey, you follow me? Uh-oh. Okay, there you go. Now we talk. So these identical twins, the minute after these two identical twins were born, if you were simply curious, then if you decide to examine these two identical twins' DNA sequence, what would be the result? Identical twins are such a clone, so it would be 100% identical. But let's say these two identical twins have lived something 100 years. And then they got reunion. Hey, brother or sister, whatever, okay, long time no see. So it's been 100 years. So shall we compare our DNA sequence after 100 years of their born? What do you think? Do you think their DNA sequence would be, again, identical 100%? Maybe, but maybe not. Because 100 years, the DNA mutation occurred separately in these two different individuals, randomly, which means in different location on their genome. So if you compare these two individuals, now there might be actually a little difference as a result of this individual random mutation occurred. But 100 years is such a too short time. So there might not be such a big noticeable difference. However, let's say these two individuals live, instead of 100 years, 10,000 years. Wow. After 10,000 years of living, and then got reunion again, and then they just decided to have this DNA sequence comparison again. What do you think? After 10,000 years? Definitely there will be some differences. But how long, for example, how long have chimpanzee and humans, the common ancestors, have separated? Probably 100,000 years or 1 million years ago. That is the reason why we had something like a little bit 2% of DNA sequence differences. So by the same logic, if we compare the DNA sequence average on an overall DNA sequence comparison from us with that of dogs, probably the differences would be something greater than 2%, obviously, something like 5% or even 10%. That means here is a common ancestor between human and chimp, but perhaps, dog, is that you? The common ancestor between human, chimpanzee, and dog would be, this is time, have existed much further in the past. Okay. So, since this separation, perhaps the DNA sequence mutation have occurred more frequently. So that is the reason why the DNA sequence difference is greater. So it is just mathematical, you can calculate. So by simply checking how much sequence similarity do we have between human or even fish, okay? So then you can draw some conclusion. Okay, so probably about how much or how many years ago we had common ancestor between human and the fishy, as opposed to some more recent past we had the common ancestor between dog and human. So that is a really convenient and very accurate way of estimating this. Totally dependent upon random event of DNA sequence mutation will present you some noticeable DNA sequence change. That is the logic. Okay. Now, having said that, then why then mitochondria and human Y chromosome? It says no recombination mitochondria DNA. what is the recombination? And I gave you this diagram. The genetic recombination occurring at the chromosomal level occurs during this particular cell division we call meiosis, during the gamete production. During the gamete production, these two chromosomes, what would you call these two chromosomes on this figure? Is there any proper name you would like to use? These two chromosomes? The what? Yeah, great. Thank you. Homologous chromosome. So the blue one probably I've got from my dad, and this red one I've got from my mom. Okay, but basically these two chromosomes are same chromosome. What do you mean by same chromosome? Does it mean that all this DNA sequence between these two homologous chromosomes are 100% identical because they are same? No, it is not. Because even if they are same kind of chromosomes, but their origins are actually different. They came from two different individuals. Your mom and your dad were not related genetically at all before they got married. Okay? So the homologous chromosome means simply the same kind of chromosome, which means on particular location. Okay, so if I draw something over here, then I'll be damned, right? So I'm not going to do that. So, but over here, that if this particular location is the location for specific gene, that means exactly corresponding same location of that homologous chromosome, you would expect that same kind of gene will be present. But not necessarily exactly same sequence of the gene. Let's say if this location of on this chromosome, if this location actually carries the gene for human eye color, for example, for instance, okay, then this exact same location, corresponding location, also would carry the gene for human eye color. However, actually, this particular gene, what kind of a trait? You can have several different options, like red, black, brown, all different kinds of human eye color as a trait you can have. So it could be black, but on the other hand, the corresponding same location, this gene will carry the trait for brown eye instead. So that's the definition of homologous chromosome. Okay. Identical, same chromosome, which means same kind of genes are there. However, the detailed traits of each particular gene could be different because mutation. As a result of mutation, variant can occur. The funny thing about this recombination is when this cell is trying to make a gamete, a sperm or an egg, then that is the only time for this homologous chromosome recognize each other. Oh, I had a partner. Where were you? How have you been? And line up. There is a very good reason why for this kind of cell division they must have this kind of ritual of finding the homologous. But anyway, they do. That is the only time. And then what happens is they have this kind of a free exchange because they are kind of same material. Yeah. Although details of DNA sequence could be different, but there's no harm. Even if you just have this kind of crossing. Yeah. After the crossing, resolving, hey, we gotta go. Huh? So let's say we have a little bit of a swap happened. Doesn't matter actually. No harm. But actually that by doing this, it is doing some benefit of increasing the DNA variation, recombination, increase the diversity of the gene, just like reshuffling your deck. When you're playing cards, every turn you have to shuffle your deck. Otherwise, the winner gets always the winning card combination, or it's not fair, kind of stuff. So that is the recombination. So recombinations, although recombination is very good, beneficial for our or entire species for the sake of increasing this diversity, but for this type of purpose, like trying to compare the evolutionary relationship accurately, this recombination is doing what? So counteractive. It's not. It will confuse. It will decrease the actual accuracy. Although overall, yeah, it is not going to affect too much of the overall result because of this recombination. However, still theoretically, it will slightly affect the accuracy. So then, if you were to find the the best material for such comparison, sequence comparison for the evolutionary estimate, then you would want to find any material, DNA material, that doesn't get any such stupid recombination. And mitochondria is the one. Why? Mitochondria DNA is not in the nucleus. Mitochondria DNA does not have any homologous DNA. Mitochondria DNA is actually haploid. Because later, probably we will have a chance to look at it, the origin of mitochondrial DNA is a prokaryote cell. Prokaryotes do not have any such diploid or homologous chromosome. They are haploid. They wouldn't. It is really safe to use as such material because they wouldn't have this type of gene recombination before they go into this DNA replication. So that's very good material. So no recombination means for this type of purpose, sequence differences, if you want to compare this sequence difference or variation, that's better becoming strictly from the result of DNA mutation, and that would be the case mostly in the case of mitochondrial DNA. Because, hey, why this one is not? Because they don't have this type of homologous chromosome. And another little benefit of the mitochondrial DNA is in a single mitochondria in a cell, the copy number of this mitochondrial DNA, multiple copy, this is just a replica, is multiple. If you go back to this diagram, in a single mitochondria, how many mitochondrial DNA do you see? 1, 2, 3, 4, 5, 6, 7, 8, or something like. And another thing is, in a single cell, how many mitochondria do you think you have? Probably something like minimum 50 or 40, 20, maximum 500, even in some cells, like even 1,000 copies of mitochondria in a single cell, depending on the metabolic activity of the cell. It varies, but bottom line is you have many copies of mitochondrial DNA. So the more the material, the better you can have the result of this analysis. So that's good. Especially if your task has to go through this scraping some of the fossil bones and get the DNA material, the mitochondrial DNA would be really beneficial because you can get, relatively speaking, you can have many copy numbers of this. And also, mitochondrial DNA gets inherited from mother to. So even me as a male, all my mitochondrial DNA I've got from my mom only. My dad's mitochondria, once they might have come in, of course, during those fertilization, but all those paternal origin of mitochondria are actively get killed in the fertilized egg for some reason. So all in animals, even most of these plants also do have this similar thing. So mitochondrial DNA for some reason always we get inherited from maternal source. In other words, if we simply follow the mitochondrial DNA, you can trace only this female origin. So if you are particularly interested in only this female origin, then the mitochondrial DNA would be the best material. Right? So there was even the name of project, was it? Yeah, Mitochondrial Eve project, like biblical. So trying to figure out what was the DNA status of these our original ancestral females, and it turned out that actually all these female living on these days, of course, outside of Africa, of course, that means outside of Africa, all those presently living these humans were all originated from a small number of females from Africa. Yes? Yes.
인간이나 그런 포유류가 아닌 포유류에서 미토콘드리아 유전자 상태였는데
Yes.
애니멀에서 콘드리아 유전자 난 사례가 있나요? 그리고
In animal?
이거 애니멀이 아닌 다른
In plants, the mitochondria, the maternal origin of mitochondria is much less common. But still, there are some kind of trend exists even in the case of the plants too. In plants, there are also like chloroplast DNA origin also goes with this maternal origin too. All these plastid. Are you listening? So all those plastid mitochondria and chloroplast origins, for some reason, overall generally follow this maternal inheritance. Although in plants there are more exemptions than the case of animal do exist, but overall trends is such that, yes. But why? If you're asking why, we can only speculate. Like, by doing so, we can have, because egg cell has much ample cytoplasmic material, as opposed to sperm only have very few cytoplasmic content. So energetically speaking, if you provide the mitochondrial chloroplast from your maternal source, then you probably have a better advantage in terms of energetics. You can have a little bit of an advantage, but that's always your speculation. No one could prove. Okay? Another thing is human Y chromosome is for the same logic. Human Y chromosome also, even though human Y chromosome is a nuclear chromosomal DNA, but because of this shape, like for example, if you try to compare the Y chromosome in this frog, like lower animal, Y chromosome, X chromosomes are actually true homologs. However, in higher mammals, like humans and any other mammals, Y chromosome and X chromosome, if you compare, wow, X chromosome and Y chromosome, although they are kind of a pseudo homolog, which means during this meiosis they align together, okay, as if they were homologous chromosome. But in reality, they are not. The number of gene and kinds of gene present between Y and X chromosomes are totally different. And even if you simply look at it, Y chromosome is such a pathetic, small, doesn't even contain many genes, all useless gene, except only one gene which determines the sexuality of whether it's going to be a male or female is determined by Y chromosome. But other than that, not many meaningful genes are present in Y chromosome. On the other hand, X chromosome, there are many important genes are present. These two genes are not homologous at all. So they cannot have any recombination occur. In other words, Y chromosomes are left alone. If you are female having two X chromosomes, you can have regular recombination between these two copies of X chromosome. But if you are, unfortunately, if you happen to be the other sex, like a male, something like male, then you are, unfortunately, doomed. You cannot have any recombination. So, yeah, that's poor thing. However, poor thing is poor thing, but just Y chromosome per se is another such an ideal material for comparing such evolutionary relationship. Especially on this occasion, if you were, for some reason, I don't think anybody would be really interested in male origin, but anyway, if you were somehow weird enough to be interested in such male origin, then you can choose this Y chromosome to compare. Then you can follow the Y chromosome Adam project to figure out what was the origin of the oldest males living. Yeah, and the result indicated similar things. All those male living outside of Africa nowadays, their ancestors came from Africa, and the number of the ancestors was very few, small number of individuals were our ancestors. Also, many interesting also the facts have also revealed, but yeah, I wouldn't really reveal right now. But anyway, that's the thing. So finally, using this DNA sequence comparison and with the advanced, very impressive advancement of the technology. So relatively recently, even the DNA recovering the entire chromosomal DNA. I'm not talking about this mitochondrial DNA anymore. So entire human chromosome of Neanderthals have been recovered. And then this sequencing have been made within relatively very small duration of this period, only 4 years. It took only 4 years. Originally, the Human Genome Project took more than 10 years, actually. It was actually the real completion of the Human Genome Project, which actually began in 1990, was, I guess, actually finished, totally accomplished in even last year. People kept it secret, but in reality, it was only done because of Y chromosome. Y chromosome was the problem. It was really, really difficult to correctly sequence the entire Y chromosome, so that's why it took really such a long time. But anyway, this Neanderthal Genome Project was completed, and then it turned out that this Neanderthal genome was also very similar to that of our ancestor, modern humans. So it has only about 3 billion base pairs, very similar to. So actually, it was the sequence identity between human and Neanderthal was something about 98.8% similarity, almost 99% identity exists. Okay. So almost the same. And more importantly, the interbreeding. Someone asked in the script paper, 쪽지, that what's the difference between Neanderthal and humans, and their anatomical difference was like this. Our direct human ancestral form was a little bit taller. Neanderthal was a little bit shorter, and their skulls. Which one is a human and which one is a Neanderthal? I don't know. Modern Neanderthal. I think the bigger cranial, probably this one was a Neanderthal, I guess. So they even had a bigger, larger brain than our ancestor. So intellectually speaking, they were not really more stupid than we are. We are already stupid enough, so I don't really expect they were more stupid. And as you have already know that this interbreeding is the thing. So gene exchange, having the evidence of the gene exchange between Neanderthal and human were made. Of course, Neanderthals were soon get extinct, so the degree of gene exchange only lasted such, relatively speaking, brief period. So afterwards, we probably must have diluted out the traces of evidence of this Neanderthal genes from our whole gene pool, if you know what I mean. But still, well, we were able to identify, okay, some portion of the genes actually we got from Neanderthals, since we have finished sequencing the entire original Neanderthal genome from their fossils. It was a really impressive one. Not only the Neanderthals, but the humans, other relative humans, like Denisovans and hobbits, Homo floresiensis and Heidelbergians, it's how to pronounce, but those other humans, related humans, found their fossils were found all over the world. They also show the evidence of this interbreeding in terms of their DNA sequences. But, okay, someone also asked that how the humans and Neanderthals migrated out of Africa at the time, because the Europe and Africa was cut up. No, it's not. If you think about it. This area, there is still land bridge, so you could migrate. You can walk from Africa to walk to the tip of Europe. So that's how you could migrate out of Africa. So that's not an issue. So anyway, many genes, a fair number of genes were actually could be traced back to the Neanderthal origin in these days. Namely, like some of the very interesting genes, like genes responsible for causing depression, allergy, and heart problems, circulatory system, diabetes, and addictions. All these all bad genes, all we could trace back to Neanderthals. So do you feel good about this fact now? Because if you are suffering from one of these genes, at least you could now blame not you but on Neanderthals. That's not my problem. That was Neanderthals' problem. So what's the takeaway of this finding? What do you think? You had like substantial portion of the genes you got from Neanderthals, and some of them are, how come all these genes that allegedly we got from Neanderthals are all such a bad genes? What's the, how should we interpret this? Does that mean, okay, so now I understand why Neanderthals got extinct and we were the one who finally survived, because we were the one who carried the better gene, and they are the inferior ones who carried the bad genes. Is that kind of general like interpretation which is correct? What do you think? Or are there any other aspects that you could view this from other perspective? So definitely this means that, okay, we were better than them. If you are kind of a innate, politically correct mindset individual, probably you would, no, I don't want to say it like this because that's not politically correct. But then offer me any logical explanation as that is not the case. Hey. So are we all okay? Agree that, okay, that means that this indicates that Neanderthals were the bad gene carrier who were primitive, inferior to us. Yes? No? If say no, then offer me other version of the explanation for this interpretation. Yeah, me too. I'd like to also say no, they shouldn't be. Neanderthals are also human. How can they be like inferior to us? What's the evidence? This one is an evidence? I don't know. There's no answer on this. Yeah.
What kind of urinary tract odors?
Gallbladder and urinary tract. You don't know what the urinary tract is. Those organs that aid in when you go to the bathroom. You have to take a pee, right?
Likely 요실금?
It could be or something else. Right? Many all kind of different things. Yes. Okay, it's an environment. Yes.
Likely 요실금. Couldn't understand.
Similar things, but if you travel, try to make a like imaginary time travel back to those days of when Neanderthals were around. Okay, so what do you think? I mean, depression. Yeah, they had certain genes. Nowadays, causing depression. Okay. On what occasion you usually get a depression? Like when you are confined in this type of a shitty room with a less light environment or some other, when you have some uncomfortable settings. But do you really think the Neanderthals were also in the exactly same environment as we do live right now? Probably not. Yeah, but their perception of stress must have been a little bit different because they were living in different environments. Allergy, for example, probably allergy would be a better example to refute this. Allergy is what? It's an overreaction to certain overreactive immune response is the allergy basically. Okay, so in these days, allergies are such a bad thing. However, if you had such a sensitive, overreactive immune system back then in those Neanderthal era times, don't you think that would have been really huge plus benefit? Because they don't have any antibiotics, any drugs. So if you have a much stronger sensitive immune response, probably that would have increased the chance of survival. But that exactly same gene we got inherited in these days where like you really don't have to have such a overreaction to anything. Now we have in the form of allergy in these days, probably. And diabetes also. Back then, probably obtaining good sugar source might have been very, very rare thing. These days, sugary, nutrient thing, candies and bread, all those goody things are plenty, abundant. So you shouldn't anticipate those Neanderthals with the same kind of a set of genes would respond exactly identical to what we would do in these days with this kind of genes. So their environment would have been totally different. And the reason why those majority of Neanderthals still carried all this type of genes that cause many problems in these days, but probably back then, those genes were doing okay, at least, minimum. Or maybe some of the genes might have actually played beneficial role. It's probably a better version of explanation. Because otherwise, these genes carriers, those inferior gene carriers, would have been eliminated out soon during the course of their evolution, natural selection. But they carried, they survived. Means probably they were able to adapt to specific environment. Yes?
그러면 내가 심장마비 걸리거나 그 당뇨병에 걸린 거 어떻게 보면 네안데르탈의 종양 때문인가요?
Not really. Because a lot of those diabetes genes we obtained through our bad habit. In this modern day bad habit, we lost the sensitivity and fine tuning ability to this sugar concentration in our blood. That's how we suffer from all this diabetes, for example. So not necessarily, or we call finger pointing all our misbehavior to them, I guess. Okay, we are again nothing new, running out of time. So let's quickly go over. Probably that's better. Where, yeah. Do everyone have this copy? Yeah, so that I can go over on which area we need to take a look at. So when you try to study on your own home, probably this thing, you might know at least this only minimum these things you need to take a look at for your midterm exam is what I'm going to go over right now. So no. Why this thing is behaving so stupidly? I'm not going to. Yeah, all kind of different chemistry. Vast majority of them. If you are major or chemistry major or biology major or physics major, you already know this, so you don't need to go over again. The problem is if you are a Mun과 student and you have never seen these things before, you are lucky because you didn't have to look at it. But so if you have, like I said, the only reason why I still putting up this for your study material is because without knowing this basic, it will be very, very superficial to understand the following chapter contents of the cells and all those internal content of the cell and their function. So at least the minimum, let's try to understand some basics so that seemingly we are pretending that everybody here is on the same page is the purpose. So having said that, first off, I'd like you to understand the concept of this radioisotope. Why this radioisotope appears on the biology course. So for example, how radioisotope can be used in biology in many different ways. But that's not something that I want to go over in details, because you can go over on your own later if you have extra time. But here, one point is, it's called radioactive dating, carbon dating, for example. So using the radioactive isotope, you can estimate the approximate time, how old is this specimen is. It's called the radioactive dating, or for that matter, in biology, usually the most commonly used isotope is a carbon 14. So sometimes it's called the carbon dating. Okay. So this carbon dating, radioactive dating, but one thing is, if I ask you, why would this carbon dating can only be applied to the non-living object, the object that already dead now? In other words, what happened if we use this carbon dating to estimate the how old are you? Let's say, assume that how old are you? If I ask you, then you'll say, I'm 100 years old, and then I don't believe it. I don't trust you. So then I will just prove it by using this carbon dating. So inject, hey, eat this, and inject C14 radioisotope carbon to you, and then try to estimate exact actually how old are you. Is that kind of thing possible? Why not? Yeah, it's not possible. It's impossible to use carbon dating, radioisotope dating for living organism. They have to die, at least. If you want to use that carbon dating, the material should have been dead already for a long time. Why? What is the reason? What's the reason why? Why do you think this carbon dating cannot be used for living object? If you can answer, then you can understand the principle of this carbon dating. That's why. Decaying. Yeah, the isotope, radioactive isotope, unlike the regular element, radioactive isotope shows characteristics so-called decay. So let's say when 1 gram of carbon C14, radioactive C14 exists. Okay, in order for this 1 gram of C14 to change into 0.5 gram of radioactive C14, it will take approximately 5,000 years. Then after 5,000 years, you have in your hand, originally you had 1 gram of the C14 radioactive, then you have 1.5 gram, half gram of C14 radioactive, and half gram of regular, non-radioactive, although it is not exactly C14, but regular material, standard material. So that's what we call the decay, radioactive isotope decay. And that is something that parameter that we use to estimate the approximate time of this object, how long this object has been dead. Why? It's much more difficult than I expected, probably, to go over this thing. Let's say there are 3 different kinds of carbon. One is regular, carbon 12. The other one is 13, and C14. Although there are several others, but let's say there are only 3 different kind of carbon exist on Earth. Okay. So C12 is the vast majority, something like 98% exists everywhere. Everywhere you look at, most of the carbons are C12. Very rare, let's say something 2%, that's a lie. Even less than 1%, but let's say for the sake of argument. And let's say this is something 1% and 0.01%, whatever. So there is some specific ratio already exists among these different kinds of carbons. And on this occasion, this is the only radioactive isotope. And this is the only one who presents this decay characteristics. So let's say during a person, let's say you discovered a dead person on the Alps, snowy Alps. You have no idea about how old this whole frozen dead body on the Alps mountain. Probably, maybe this person has been dead for 10 years. Well, you don't even know. It's been dead for like 5,000 years or maybe even 10,000 years. How do you know? How can you estimate correctly using this method, carbon decay? Now, when this person was alive, all this carbon, through metabolism, would have been circulating. So, the inside of this person's body, the ratio of all these different carbons would have been identical to the standard regular ratio of the whole entire Earth's carbon ratio. But once this person is dead, ticking, then what happened? All this metabolic circulation has been cut off. So at that time, at the time of the death, any remaining carbon inside of this body will be trapped. Okay? So then let's say 5,000 years later, this body was discovered. And if you compare the ratio of this carbon, what do you think? All these carbons, we don't have any way to figure out whether this carbon's ratio was something. However, one big difference you can immediately notice the difference in radioactive isotope. Why? Because inside of this body, this radioactive isotope would have been decaying over time accurately. So like if there was a 5,000 years, then if originally this C14 was present in the same ratio, equal ratio to those of the outside, but you would find only half the amount of C14 inside of this body because the other half would have been gone through decaying. So that's how you can estimate the relative time after this person or these animals or these plants death. So if I ask you, for example, on the exam, why this carbon dating or radioisotope dating cannot be applied to a living person or a living organism, then what would be your answer? Of course, I'm going to give you an example, multiple choice examples. I'm not going to let you write down a free essay about that. You should be able to identify correct answer. Okay? So that's one thing. That's one area that you might want to take a look at about this. And the second thing is, why carbons? Why in organic molecules? In organic macromolecule, why carbons presence of carbons are so important is another thing you might want to understand. The carbons, importance of a carbon in forming biologically important macromolecules. This is another. And another thing is, should be able to distinguish the difference between. There are 4 different types of chemical bonds have been introduced into the chapter. One is covalent bond. The other one is hydrogen bond, ionic bond, van der Waals bond, the ionic interaction, the hydrophobic interaction. But anyway, 2 big things. One is covalent bond. And another all different kinds of bonds we collectively called a non-covalent bond. Tell you what, this covalent bond is very strong, stable. All these non-covalent bonds like hydrogen bond, ionic bond, van der Waals bond, they are all very weak bonds. But these chemical bonds are also biologically very important, especially in enzyme substrate reactions, antibody, antigen recognition and binding type of, all very finite interactions occurring in biology. All these non-covalent bonds are very important. Covalent bond is very strong, containing strong, huge amount of energy. Okay? So all these big macromolecules, your foods and all kind of the macromolecules are all built through covalent bond. On the other hand, these all non-covalent bonds are very weak bonds, but still, biologically speaking, many activities, biologically important activities are also dependent on this non-covalent bond. Why? That also could be on the exam. For example, I will specifically give an example. What would happen if enzyme substrate interaction, in these days currently, enzyme substrate interactions are dependent upon this non-covalent weak bond. But what would happen if a certain enzyme, or particular enzyme, is dependent upon this strong, stable covalent bond for its activity? What would happen? Would it be good thing or bad thing? Have no idea? This weak bond, although strength of this bond is so weak, however, because they are weak, they can be very temporally flexible, so it can be done. It can change its status very rapidly. So in other words, if enzyme interaction with the substrate had been dependent upon covalent bond, the speed of this enzyme catalysis would have been much slower. Because you have such a strong interaction as a chemical bond, so in order to have a quick change after the completion of the chemical reaction, if you want to have another substrate, the time it takes would be much longer than current this status. Okay. I don't think just by Okay, for that two question would be definitely on the exam and probably two or three more question if I want to. I haven't decided exactly that will happen or not. But right now I'm just kind of roughly thinking there might be two or three more possibly, or maybe only two of these will be on your midterm exam. I don't know. But if any other questions regarding concerning this chapter are to be on your midterm exam, I'm going to also like instead of going over this lecture, I don't see any really positive reaction from the look of you by just going over these things. Probably not many of you can actually follow this, so I will just stop. If I have more of this kind of questions regarding this, then I'm going to specifically post on the snowboard. Okay, this particular question will be on in the form of previous exam questions. Okay. Someone asked about, am I going to give you some previous sample exam questions before the exam? Yes, I will. I'm going to put some previous exam questions, midterm exam and final exam too, on the snowboard. However, one catch is I may not have exactly the same type of questions on this coming exam. So you cannot really bet on entirely onto that one basket for your studying exam. So that's what I'm going to do. So I will stop doing this, and then next lecture I'm going to jump right into the new chapter, which is a cell. And whenever I find necessary, I will try to bring up some specific one about this background, the chemistry. Probably that's a better way of to lead everyone. Try to put everyone on the same page, I guess. Okay, so Wednesday we are not going to have any lecture, so see you on next Monday.